RNA, silencing RNA. Is anybody in here taking biochem yet? Where you're in the biology biochem, Brother Cannon, would you mind? Okay, good. So hopefully we're going to be able
Before
to get that
siRNAs.
background. But, a lot of times you go to presentations that are way over your head, and it's oftentimes during those experiences that you begin to either glean information that's significant or recognize that you need to study harder, right? So this will be a good experience for you. So, turn the time over to Teresa.
Okay, so, well, my project was about siRNAs. So I guess first is probably none of you know what an siRNA is. What it is, it's a small interfering RNA. So it's basically a little molecule of double-stranded RNA, which is unusual because RNAs don't usually like to be double-stranded. But these are about 21 to 23 base pairs long, and they have a little 3' overhang of 2 nucleic acids. And they're involved in a mechanism of gene silencing. So the whole thing is called RNA interference. And it's actually pretty cool. So what happens is, this is actually a process that's present naturally in eukaryotic cells. There's a protein in your cells called dicer, and it cleaves the large double-stranded RNA molecules to form these little siRNAs and microRNAs. And then what they do is they join with these proteins to form a complex called RISC, which stands for RNA-induced silencing complex. And then that complex goes and it uses the siRNA to find mRNAs and cleave them. So if you don't know, mRNA is the precursor to a protein. It's basically the template that a protein is made from. So some of the uses of these, they're very powerful in gene studies because they can be made in a lab. So you don't have to make them in a cell. You can make them in a lab just the way they are and transfect them into a cell pretty easily. And you can make them against just about any gene, as long as that gene has a complementary sequence to your siRNA. If you put that in a cell, it's going to find the mRNA and it's going to cleave it and it's going to knock down the expression of that gene. So then they use them to study genes, like I said, and more recently they're actually trying to use them in therapies. So ways to knock down different genes to help manage diseases. And so here's just a little diagram of how it works. So you have your double-stranded mRNA, double-stranded RNA, and your dicer protein cleaves it into a little section, and then it joins with the proteins that are called argonauts to form RISC. And then RISC uses the sequence of your siRNA to find the complementary mRNA, and then it cleaves it so that it is degraded and cannot make proteins. And so then the scope of this project, we had 4 siRNAs in the lab. So I decided, and they're all against the protein GFP, which is a really cool little protein that makes things glow green. And so the thing was that all of the siRNAs, they were all purchased from companies except for one that was made here. But we wanted to see if they would have different efficiencies in how much GFP they were able to knock down. Because one of the problems with making siRNAs for use in laboratories is that the target sequence affects how efficiently it works, even for siRNAs against the same gene. But they're not really sure how or why that happens, so they're not exactly sure what the parameters are that make it an efficient one versus a non-efficient one. So that's one of the problems. So this, if we can tell the difference between these four, it just adds a little bit of data to figuring that out. So the methods that we use then, we basically did two. One of them was just visual and one of them was quantitative. So for the visual, we use fluorescent microscopy, and it's based on the fact that GFP is bright green. So we transfected cells with with respective siRNAs and a GFP expressing plasmid. And then we let it grow for a few days, or we let it express for a few days. And then we just visualize them under a fluorescent microscope. So then we could see how many of the cells were green, how much green was present, and that would give us an indication of how much GFP was in those cells. And then the other method was Western blotting, which is when we isolate all the proteins in the cell and we run them in gel electrophoresis on a polyacrylamide gel. And then what you can do with that is you transfer them to a membrane and you probe them with an antibody that's only against your protein, the one that you want to detect. So then the antibody will bind to that protein, and in this method there were two antibodies, so there's the primary one that binds the protein, and there's a secondary that binds the primary, and that has a conjugate. And then when you add that conjugate, it makes a little luminescence that you can detect in bands. So that was what we did. And then you can analyze the intensity of those bands as an indication of how much there is. So then these are the results for the microscopy. So as you can see, this is the positive control that had only GFP and no siRNA. And then we can see this is the one. So the four siRNAs that we used were called Thermo, Invitrogen, Ambion, and then the other one that was synthesized here, I've named in this lab just BYUH. So then we can see that the first two, there's almost nothing there. That one, if you look really hard, you can see like a few green, like-ish blobs. And then these two is less than that one, but there's still a significant amount. And then that one is the negative control, which had nothing. So then from this, we can see that there definitely is a trend that they don't all work the same. So then, to get our quantitative results, I ran two different trials. So the green bar is the first trial, and the red bar is the second trial. And what this is a percentage of expression based on the control. So it's basically the intensity of the GFP from each different siRNA over the intensity of the control band. So then we can see for these ones, the Thermo and the Invitrogen, they're both very, very low. We have most of the GFP was knocked out. And then for the Ambion and the BYUH, there is a significant amount. I'm not sure why on the second trial there was all the percentages were lower. I haven't figured that out yet because I just did it this morning. But as you can see, there is the same trend. So the conclusions that we're able to make is that the Thermo and the Invitrogen siRNAs are definitely more efficient than the ones from Ambion and BYUH, but we still haven't been able to figure out definitively which one out of those two groupings is better. Going along, the Thermo, in all of the tests and trials that we ran, the Thermo was the most efficient, but we haven't been able, the numbers that we have so far didn't show it. So we would need to run more trials. And then there were some problems with the Ambion and BYUH sometimes. There was fluctuation in which one had more expression. So that needs to be addressed. So those are only 2 trials, and those are the only 2 that I had time to do so far, but if we did more, we could probably figure out conclusively like which one, we could put them all in order, which is the ultimate goal. And then the only thing is that we're not sure, we don't have a way to know at this point if the difference in their expression levels was actually because of their target sequence, or if it was because there was just problems with the stock solutions of RNA that we used. So, yeah, that's. Does anyone have any questions?
So do you think if you tried the same thing with a different gene, do you think you would get similar results?
Well, we couldn't use those same siRNAs.
Obviously,
Well, yeah.
but try the same procedure, order the siRNAs for, glutathione reductase or some other enzyme and tested the activity. Do you think you'd see similar results that Albion ones would be less effective, or do you think that's just had to do with this particular gene?
I don't know, it might have been just because of this particular one. I found it kind of strange that the one that came from Ambion would be so inefficient. So that was why I was wondering if maybe there was a problem with our stock solution. And I think that's something to look into. But since it's just based on the target sequence for each gene, I don't know why one company would be consistently less efficient.
Be interesting to try. There's different proteins.
So this slide here is, just to make sure everybody's understanding, pictures of cells. Chinese hamster ovary cells, is that right?
Yes.
Okay. Chinese hamster ovary cells, and the cells are the green blobs. And then they're green because they're making this green protein. And the thing that you're putting in there is supposed to stop them from making the green protein, and that's why you get some black, correct? So how do you, growing these cells is kind of tricky sometimes, right? Or not really. They seem to be, you have to be very precise in when you kind of feed them and how you change them and take care of them, right? Is that not the case?
Yeah, you have to split them every 4 days.
If the cells were unhappy for any reason, would that make them behave differently?
It might, but all of these ones were run at the same time from the same, they were all split from the same stock. So if there were any discrepancies, it should have been for all of them.
And you must have to control for concentration somehow, right? For the cells on the surfaces. How do you do that?
That's why we use the same amount. So when we split the cells, we use the same amount for every well. And we assume that it's more or less homogeneous once we trypsinized it. So there should be about the same amount of cells, and then we can check it with the microscope to make sure.
I think it's your second to last slide or something up there. That's your Western blot results, right? I'm
Yeah.
sorry, right there. So on the Ambien and the BYUH, there's not a lot of inhibition. Comparatively, but what is the amount? Did you guys run a Western blot for just your GFP expressing control, your positive control?
Yes, we just didn't display it. So
Okay.
this is, what this is actually a percent based on the intensity of the positive control.
Okay, perfect. Thank you.
All right, are there any other questions?
So if you scaled up, sorry, one more question. If you scaled up, for example, that thermo results from the second run, if you scaled that up to be the same as the first run, multiply it by, what, 3 or something like that? Is it about a third?
I think so.
Would the Albion and BYUH results line up with their trial 1 results as well? Or is it significantly less?
I'm not sure, I haven't looked into that.
You should look at, just multiply them all by the same factor and see if they
Yeah.
have a similar.
I only did the second trial today, so I was just, haven't organized it yet. But then there's also problems with the Ambion and BYUH because I think that there may have been a problem in that trial with the actual pipetting because the results were kind of strange for both because this is an average of 2 samples. So the samples for each of them were very different. So that was part of the reason why I wanted to run more trials to
So
clear
they're
that
gonna
up.
stay on the semester to do more trials, right?
I wish. Not really.
All right, let's thank Teresa.